Okay, so next part is we're going to look at absorption. Absorption in the small intestine, okay? So let's go back all the way through all this stuff. Oh my gosh. And we see, remember, we started chemical digestion. Now we've broken down into glucose, amino acids, glycerol plus fatty acid chains, okay? So in the jejunum and ileum, they also have these circular folds. So remember, these like bumps are the circular folds. On every circular fold, you have a villi. So this is my villi. The villi are the finger-like projections that project out of that circular fold. And then on the villi, you have cells, epithelial cells, and those epithelial cells have their own tiny little microvilli. Okay, I'm just going to draw one really big cell with microvilli. Okay, it's not that big, but it's helpful to draw it bigger so that you can see what's happening. Okay, so that's my cell plus my microvilli. Remember, all of this stuff increases surface area for absorption. The chyme is going to get stuck in all these crevices, and as it gets stuck in all these crevices, it's gonna have more opportunity for these small molecules to actually be absorbed through the cell layer into the various parts of the body. So what we're going to see is we are going to have an artery here, and this artery is superior mesenteric artery. If you don't know why, you should figure out why. Okay. And then we see that arteries and veins actually are connected together through a capillary system. But I'm just going to draw my vein here, my superior mesenteric vein, which is going to go to my hepatic portal vein, like that. Again, if you don't know why this is superior mesenteric vein, you should probably figure out why. Okay. And then the green part here, this is the lactule. The lactule is part of the lymphatic system, and eventually it actually dumps into a vein. The hepatic portal vein, no. But eventually it's going to connect back to the vein system. I need 2 out of 3 of these things. Veins pick things up, and so do the lactule, okay? So what I have to do is I have to kind of go through chemical digestion in order to figure out what I'm absorbing. So let's say I'm going to have a carbohydrate. The carb is going to be broken down into glucose. Which is pretty small. It's like that. And then I'm going to have a protein. Proteins are going to be broken down into amino acids, which are going to be like about that. Then I can have lipids, okay? If I have lipids that get cut by lipase, I'm going to have really small lipids, or I'm going to have small fats. Again, kind of maybe like that. Okay? This is going to be sort of easy, sort of. Glucose will just simply go through the cell, and when it goes through the cell, it gets absorbed into the vein. When amino acids go through the cell, again, they get absorbed directly into the vein. Small fats can also go directly through the cell, and then they also get absorbed into the vein. Really into the capillary, but it's going to move out of the body like that direction to the hepatic portal vein. Okay? This is pretty straightforward. Anything super small can go through the cell membrane layer and gets absorbed into the vein. But sometimes fats don't break down all the way. Sometimes the lipid breaks down into like glycerol plus 1 fatty acid. And then maybe like additional fatty acids. And these are kind of big, like you have a glycerol and a fatty acid, and like you have fatty acids. Well, those are still kind of big, so we didn't have time to cut them. And since they're still primarily fatty, how do we transport that towards this cell and then get it stuck in the cell? To do this, I'm going to take and I'm going to surround all this with bile, the bile salts. And that ends up forming my micelle. Okay. My micelle is going to be transported right here. So I transport it towards my cell, and then the bile salts are going to disintegrate away. And my bigger fatty acids here are close enough to the cell that they can get absorbed. It's like, why can they get absorbed if they're so big? Well, remember the fatty acid, the cell membrane is made out of fats. So fats can actually absorb other fats relatively easily. The problem is most of the inside component of the cell is water. So now you have fat inside water and they're going to want to push against each other. So what happens is once this gets inside the cell, it gets put back together as a triglyceride. But that triglyceride is fat inside of a water-based area. So we're going to surround that with protein. And this is called the chylomicron. Okay, the chylomicron is going to get pushed out of the cell. But it's so big that as it starts to move towards the vein, I know it looks big here, but the lacteal will be much bigger and have these little openings. It actually gets sucked up into the lacteal instead. So the chylomicron is going to have these bigger lipids, these triglycerides that get absorbed better into the lacteal. And then as you get to bigger veins, you can dump those fats back into the vein system because the vein will be much bigger now, okay? So you want to understand lipids that are much larger are going to have to be transported by the micelle towards the cells. The cell then reconstitutes or remakes the triglyceride, surrounds it with protein to create a chylomicron, and it's the chylomicron that gets absorbed and transported via the lacteal, okay? So hopefully that helps you understand how absorption occurs in the jejunum and the ileum a little bit better. It might be more understandable when we get to blood vessels because you'll understand the shape of the blood vessels. You'll understand how small they are and how they can absorb really very large things. So really big fats can't be absorbed by a vein. That's why the lacteal is more important. Okay, so that's it for small intestine stuff. We just got one more part to do, which is the large intestine stuff.